Facebook group and put up there that may be of interest to you. So I would encourage you.
So I locked my keys in my car right before I walked in here.
It's like the longest trail off the street chatter I've ever
Campus police get fix that, or am I gonna have to call off? You're gonna have to call off.
It's firing back up. Hey guys, class is starting. Among other reasons to join the Facebook group is I'm going to start posting these things because these summer research programs are starting to come into my email inbox. So they're places that we've either sent students or students have applied, so they got my email address. For example, if you wanted to spend the summer in beautiful, scenic Lincoln, Nebraska, they're all very competitive, so there's nothing wrong with Nebraska. The University of Nebraska has it's not an NSF program, so you won't find it on the REU list I talked about on Monday. It's through their, I don't know who funds it, but maybe perhaps the university. And then another one I wanted to point out was the summer program at Emory. So if you have to stay local, there are worse fates than spending a summer at one of the leading universities in the world. Emory also has another program. It's not an NSF funded program, but they pay you, put you up at Emory, and let you work in the research lab. They did offer an LCA opportunity for finding errors in the textbook, and one student found a typo, and I meant more science, but I decided to give her 2 points anyway. So 5 points for science error. There was one in chapter 3. Nobody has found it yet. Up for grab. And first one to post it to Facebook wins. Yes. I'm sure there are other errors elsewhere, but the one that irritated me the most is in that. Since that's what we read today. One other thing, the quizzes have to be done by 1:30. I had several before class, and several students emailed quizzes 1 and 2 well after. And I'm going to be a jerk and not give you any points for it. A little team quiz. I don't want to go down the slippery slope. So sorry to those of you who emailed them late. Just do them on time. 2 other points about the quizzes. When you take your screenshot, get the chapter title on there for me, please. What was the other thing? Oh, first, as of 12:09 p.m., the current LCA totals are up on D2L. I think I've got about 4 quizzes in. In the next 4 minutes, those are not reflected in the total. You can start following along, which will help those of you who are paranoid about your LCA totals give you something to do. Okay, and if you think I've made an error, let me know. So finishing up chapter 2, we had done detection of viruses. We talked about infectivity, electron microscopy, and detection of antigens. And I very briefly, perhaps too quickly, went over ELISA, which is enzyme-linked immunosorbent assay. So that's all I wanted to talk about. Anyway, international symbol for antibody. There are other uses for antibodies, and generally we're going to have some sort of a probe covalently attached to the antibody, and that's certainly the case if you're going to use them in the other protein level detections, which is confocal or fluorescence microscopy. That has to be, not surprisingly, fluorescent. And then there are other ones. The one near and dear to my heart is optical biosensing. Neither here nor there with respect to the course, but that would give us the ability to detect viruses by attaching an antibody to a surface, and then when the virus binds, seeing a signal in an instrument. And then I'll let you in on a little joke here as we go along. There's Western blotting, which is running out an SDS page gel, transferring the proteins over to a membrane and probing the membrane with the antibody so that it once again, antibody binds to the viral protein and you detect it through some method. All right, that's it. Rid of a lot of dry erase markers. Okay. So number 4 on our list of detection of viruses I'm sorry, 1, 2, and 3 were electron microscopy, so imaging of viruses. 2 was the infectivity, so are they cytopathic? Can you show that they infect cells? 3 is detection of antigens, so the use of antibodies or any number of methods. The example in your text is ELISA, and that's what I'm most likely to ask you about on a test. There are plenty of other uses of antibodies binding to antigens via techniques, microscopy, kinetic binding methods, and imaging off of SDS gels. And then 4 is detection of nucleic acids. One of the crappy things about being a scientist is you get really mad during science fiction shows. Back in my day, there was this show called The X-Files. Everybody says my wife looks like Scully. The characters. And she was infected by an alien virus and went and did a Western blot on her own cells. And it really pissed me off because I was in the dark days. Actually, I was in graduate school at the time, so I wasn't that good. But she did a Western blot in like 20 minutes. It really takes like a day and a half. Anyway, we also have similar methods with nucleic acids. So this is DNA, RNA, of course. We'll talk more about the structure in a few minutes. Hybridization. So one of the distinctions I'll make with respect to viral biology and viral structure is the biomolecules matter, and they're different. I'm going to juxtapose proteins and nucleic acids. Proteins do the work. Nucleic acids carry the information. And we'll talk more about why when we get there. Nucleic acids, from an experimental point of view, are very easy to work with compared to proteins. They're in many ways a much simpler molecule. And so synthesizing DNA and RNA is as easy as clicking a mouse and entering your credit card number. I do it all the time. It's very cheap. It's probably down to 25 cents a base. So if you know something about your virus, characterized it, you can sequence the genome, you can then design a probe that you know will hybridize, bind to a strand of viral DNA RNA. This isn't limited to viruses. This is used across all areas of biology. And you can, much like labeling that antibody, label the probe. In the olden days, it was with a radioisotope. Now it tends to be with fluorescent markers or biotin or some other way that's easily detectable. And so if you run viral DNA out on a gel and transfer it over, you can then treat it with the probe, and if your viral DNA or RNA is there, the probe will bind to it. If the probe binds to it, you've detected it. And so the techniques here are called southern blotting, and that's the DNA. And then, so the reason why it's called southern blotting is it was developed in the laboratory of a guy named Ed Southern, and biologists, being the muckety-muck yuck-yucks that they are, when the technique was applied to RNA, they named it northern blotting. That's nerd laugh. And that's why, once it was developed for proteins, that was western blotting. Just as you go through your scientific lives, know that western blotting is slang. For some reason, northern blotting is. And so it's the atomic level details that's carried within the information of that nucleic acid that allows for binding. So we know A binds T, G binds C via hydrogen bonding among the nitrogenous bases, and the order the The nucleotides within the sequence itself, over a stretch that's fairly short, makes it a unique sequence, and then it hybridizes, and if it hybridizes, you can detect it. Another method is polymerase chain reaction, which I'm going to assume you've had in prior courses. We can talk about it more if you like. Polymerase chain reaction was one of the most important technologies of the 20th century, I think it's fair to say. It allows for detection of minute quantities of nucleic acid because it's a chain reaction, so it amplifies DNA. So PCR is qualitative. With advent in technologies, we now have a number of types of PCR that all have something special about them. There is RT-PCR, which is reverse transcription polymerase chain reaction. It is quantitative. So with PCR, you can detect, do I have viral DNA in a sample? You could theoretically develop a clinical assay, someone infected with the virus, take a sample, grind up the tissue, do a polymerase chain reaction. If you get a product, and you should know what size your product will be, if you get a product, the virus is there. If you don't, it wasn't. You can assume you've done the controls. But that's a yes, no. RT gives us a quantitative, how much was there. So there's a little trick on the PCR. And there are any number of ways to do the little trick. The one I'm showing here is a proprietary one. It's called TACMan. So the original PCR DNA synthase was from Thermus aquaticus, which is TAC. So I'm just going to describe TACMan. So your TACMan probe is an oligonucleotide, a short stretch of DNA that you know hybridizes to the sequence of which you're interested. So, viral gene. It has two fluorescent molecules on the tag, one of which is excited at a wavelength that in the instrument you excite with, and it fluoresces, except that it's near in space to a quencher. So you get in RT what's called, well, there are a number of places it's used, but this is called FRET, or fluorescence resonance energy transfer. So the quantitation relies on the fact that a certain wavelength of light excites a fluorophore. Its excitation is transferred to the quencher rather than emitted as light. Fluorescence is an emission of light at a different wavelength than at which it was excited. And so as long as this molecule is intact, you don't have as much of a fluorescent signal as if it were. When your polymerase chain reaction occurs, that synthesis of the new strand eliminates a binding site for the probe. And so polymerase chain reaction, Maker doesn't have a good handle on PCR. Everybody either does or doesn't want to admit it. So the steps of PCR, of course, are denature your double-stranded DNA, anneal the primer, the oligonucleotide, and then synthesize a new strand, so that every cycle you get double the amount of nucleic acid you had there in the first place. As the original sample is amplified, there are fewer and fewer binding sites for the TACMan probe. It's outcompeted by the emergent products, and so it is displaced by synthesis, and as it's displaced, it's cleaved by an enzyme that cleaves single-stranded, and then that probe is no longer near the quencher, so you see an increase in fluorescence. So if you have a sample of viral DNA, it's going to be less fluorescent prior to amplification. As you amplify, you're going to get more of a fluorescent signal. The number of cycles, so this is what an RT-PCR data look like, the number of cycles it takes to get to a point where you see a significant fluorescent signal tells you how much nucleic acid was in your original sample. I'm sorry, I said reverse transcription, PCR. That is a type of PCR. This is real-time PCR. Sorry. RT, same abbreviation. So the number of cycles it takes to get to a higher fluorescent signal tells you how much was in the original sample. So you can do a calibration to know that X signal means X number of micrograms DNA. And so the more DNA you have in your sample, or RNA, if you're doing reverse transcription, real-time PCR, which is also possible, the fewer cycles it takes to get to that signal. So less viral, more viral, and it's quantitative to the point where you can calculate the concentration that was in your original sample. And again, there are a number of technologies for doing this. I just picked the one I know. We have this instrument here at KSU, so if you're really into virology and you'll find somebody to do a project with, you can do exactly this here. This is another infectivity assay. I'm following along with your text here. We can be quantitative with that as well. So this is showing you a viral titer. Which is taking a sample of virus and diluting it serially. Usually you go tenfold because that makes the math really easy to do. And then you have a number of samples, 96 wells. 96 wells tends to be a convenient one. And for example, you then test for infection. At less of a dilution, these pluses are all how many of your samples are positive for infection with the virus. And of course, as you dilute your sample, you get fewer and fewer, and you can do a TCID,